Okay, so speaking of classification, when I was in school, there were 5 kingdoms, and that was the biggest classification grouping. And one was called Monera, that was all the prokaryotes. And then we had the fungus, the animals, the plants, and the protists. And what happened is there were some scientists. One of them is Carl Woese, and there was another one named Fox, but Carl Woese gets most of the credit here. He did a lot of experiments looking at ribosomal RNA genes, and when he compared the sequences, especially of different bacterial types or different prokaryotic types, I should say, he saw that there was really a big split in the rRNA genes and some certainly other structures and other things, but a lot of it came from a discovery of the rRNA genes being so different within the single group of prokaryotes that he proposed, and it was agreed that we need to split into 2 separate groups of prokaryotes. So we now call them archaea and bacteria. And so he's responsible for that. And then the study of the rRNA genes from all the eukaryotes, it made sense to keep that as one group with kingdoms within the eukarya placed there. And so that's where domains first came from. That's where that classification concept came from. Okay, so we're constantly focusing on how can we identify characteristics of organisms so that we can identify them. For a medical setting, you want to come up with the identification of the pathogen as quickly as possible so that you can get to the right treatment as quickly as possible. What we've been talking about so far, these are all physical characteristics. When you look at an organism under a microscope, you're looking at the outside structures. You may be able to see the inside structures. You're using stains to tell you things about the physical characteristics. You're looking to see if they have flagella. Do they have capsules or slime layers or pili or fimbriae? And all of these physical characteristics do help you to narrow down what this pathogen could be. In addition to that, we also do, and we're going to start doing in lab, biochemical tests. So it's not an either or, it's just additional data coming from a more indirect way of testing. We're looking at the biochemistry characteristics of certain cells because certain species have molecules that other species don't. Certain species produce enzymes that other species don't. And so by testing the biochemistry of the cells, that can also be additionally used for helping to identify. Serological tests look for antigens, and antigens are things on the molecules on the surface of cells or viruses. So serological tests look for those kinds of presence or absence of those kinds of molecules. We call them antigens. We can use something called phage typing. That's particularly used to identify bacteria. Phage typing we actually won't talk about in this class too much. We have one little lab that we look at some phage. But biochemical tests, in addition to the physical characteristics, those are going to be big topics for us throughout the lab portion of this course especially. And we will do some serological tests and some phage typing, but it's a much smaller period of time that we will spend on those. And then analysis of nucleic acids. Now, I am a geneticist by training, so I am never going to be convinced that there's any better way to identify something than by analysis of the genetic material. So certainly that's on the list for me. That would be the first thing we would do. However, in this course, we're not going to be able to perform that kind of analysis, but ideally, that is a great way to identify different species of bacteria. In fact, a lot of times, remember we talked about Carl Woese, a lot of times the way you can identify bacteria is by sequencing the rRNA genes. So, but we're not going to get a chance to do that kind of analysis. We've been doing physical characteristics, making slides, stains, and so on. We're going to do a series of biochemical tests, and then we'll do a little bit of serological testing and phage typing. But this is all geared to identifying the organisms. All right, so these slides just go through what I just said. All right, the physical characteristics have to do with how they look and the microscopic level. You can also look at them on the, on agar plates and look for characteristics there, all kinds of physical characteristics. Biochemical tests, those are ones that we will run in the lab. And those are going to be based, you're going to get like color change reactions to tell you whether a certain chemical or certain biomolecule is there or not there. And so these are going to be indirect tests that can create a color change reaction to tell you the biochemistry of the organism. Serological tests, as I mentioned, and we'll talk about this much more in detail when we get there, but it really studies whether or not the organism has a particular antigen, something on the surface, okay, of the virus or the bacterium. Also, humans also have antigens. So anything on the surface of a cell can be called antigen, or on the surface of a virus. And then nucleic acid analysis is not something we're going to do, but our RNA genes are often used for this, and we can thank Carl Woese for that. All right, a dichotomous key is it's a flow chart or a decision tree that we're going to use in lab. And we're going to kind of do the old-fashioned one where it's just on paper. Usually a dichotomous key, if we're trying to identify a bacterium, it starts with, is the bacterium gram-positive or gram-negative? And then from there, it may ask about other characteristics, whether they're physical characteristics or biochemical characteristics, for example. And you can keep narrowing it down by these binary questions, yes or no questions, till you get to the answer. So this one is showing a particular dichotomous key. But typically, if you're trying to identify bacterium, is it gram-positive, yes or no? If it's a no, if it's a yes, that would lead you to a whole nother part of the decision tree. On this one, let's say it's a no, then they'd ask for the shape of the cells, so rod shape or bacillus shape. And then we look for some biochemical things. Can it tolerate oxygen? Can it ferment lactose? Can it use citric acid? So these are all biochemical kinds of things. But by this series of yes or no questions, you eventually get to the genus name. And there are even some additional ones that can get you all the way down, sometimes all the way to the very specific species. Now, if you can get to the genus, that's often good enough to come up with the right treatment, because sometimes no matter what species it is, you would have kind of the same treatment. But there are other cases where you do need to figure out the exact species. So it kind of depends on how that process goes, what you need to do next. But that's called a dichotomous key. It's just a way, kind of a yes or no flow chart where you use the results of your physical tests and your chemical tests to hopefully identify your pathogen, your organism, your unknown organism. All right. So that's the end of chapter 4.